The author, Nabil N. Jamal, once wrote, "It is not over unless you stop trying."
Welcome to the A&P Professor, a few minutes to focus on teaching human anatomy and physiology with a veteran educator and teaching mentor, your host, Kevin Patton.
Is there really nothing new to discover in human anatomy? In this episode, I'll review some recent finds. I have a question for you. Is anatomy finished? What I mean by that is, have we discovered all of the structures of the human body? Already mapped them all out? Well, of course not. There's still a lot left to be discovered in anatomy. Now, sure, it's true, we're not going to find another organ the size of the liver, or the size of the heart, or the spleen. Probably. Who knows? Maybe we will. But it seems kind of obvious that we're not going to do that. But there may be something that size, or maybe even just a little bit smaller, maybe some part of the liver or some part of the heart that even at the macroscopic level we've seen before, might even have a name. But now we realize that, well, part of this is separate from the other part structurally. That is, they have different structural characteristics and therefore probably different functions, right? Because we know that structure and function are intertwined with one another. So we make those kinds of discoveries fairly frequently in anatomy. That's true. They may not be as frequent as in physiology. I don't know that that's true, but I'm just thinking it. That's my impression. But, you know, I've heard this a few times recently. This idea that anatomy is finished. And haven't you? Haven't you even thought those thoughts? Maybe not, but I have. I don't know if I've ever said it out loud, but I've heard other people say it out loud. And I know I've thought those thoughts that compared to physiology, anatomy is kind of done, right? I mean, once you learn your anatomy, you've learned your anatomy. But of course, another part of me says, no, that can't be true because we are making new discoveries. And how do I know that? Well, because I listen to this podcast and also I do the research behind each of the segments of this podcast. And over the past 4 years that we've been doing this podcast, there are a number of anatomy discoveries that I've talked about, that I've brought to you. And so I thought, wouldn't it be fun to go back and pick out a handful of those and review them? I wanted to do it just for my own purposes because I know that when I go back and review things like that, I understand them more deeply the second or third time around. And that certainly happened as I put together this episode, because what I did was I took 7 segments from previous episodes about anatomy discoveries, recent anatomy discoveries, and I've taken those classic segments and I've put them into this episode. So the whole rest of the episode is one segment after another, one discovery after another that you may have heard before in a previous episode, but now it's time to review. So, hey, let's get going. A searchable transcript and a captioned audiogram of this episode are funded by AAA, the American Association for Anatomy. One of my favorite resources from AAA is their journal for teaching and learning anatomy and physiology. It's called Anatomical Sciences Education. You need to check it out. And you can do that at anatomy.org. Here's one of those structures that some folks have and some don't. The fabella. It's a bone, but it's not among the 206 human bones that we usually count as the standard bones of the human skeleton. So let's take a quick look at what it is and where it is. Then I want to share some recent news about this interesting little bone. The fabella has that name because it resembles a little bean. A bean is faba in Latin, and the ella suffix means little, so little bean. It's a sesamoid bone. And we know that sesamoid bones are usually little bones that may or may not develop in the tendons of muscles. The only sesamoid bone that's among the standard 206 bones that we count in the human skeleton is the patella of each lower limb. Most sesamoid bones are much smaller than the patella, and they may be present or not, which is why they're not counted in the standard 206 bones. So here's a good example of human anatomical variation. Variation from what? Well, from an arbitrary, perhaps even mythical ideal of a typical human. Really, it's all about numbers. And I'm not sure any one person can identify themselves as, quote, normal or typical. But a lot of us are pretty darn close. And anyway, we have to start somewhere in our understanding of anatomy, right? So why not start smack dab in the middle of a spectrum of variation? Sesamoid bones are called that because many of them are tiny little bones that resemble sesame seeds. These form in the tendons of muscles and could have any of several functions, like helping reduce friction within the tendon, helping redirect the force of muscle contraction, maybe helping leverage the mechanical force of muscles, like what happens in a patella or kneecap. Or sesamoid bones may just show up and have no function at all. Or worse, they could cause pain or get in the way of normal function. So getting back to the fabella, this sesamoid bone sometimes develops in the tendon of the lateral head of the gastrocnemius muscle, just posterior to the lateral condyle of the femur. Some biologists think the fabella can act like a kneecap, helping increase mechanical force of muscles. At least in some primates, that's what happens. Well, there are certain old world monkeys that do this. But perhaps also including our pre-human ancestors. But we sort of lost it along the way of evolution, with it showing up only rarely now and again as a sort of extra little bone, like the others that may show up in some individuals. But some recent news has some folks thinking that maybe the fabella is making an evolutionary comeback. That's because it has become 3 times more common than just 100 years ago, jumping from, about 11% of the population having 1 or 2 fabellae to up to 39% occurrence in the human population. So 11% to 39%, that's a significant jump. I know, some of us may be thinking, can evolution really happen that fast? 100 years? Well, if you're thinking that, you didn't read that book on evolution that I recommended you read in the A&P Professor book club a few months ago. It's called Improbable Destinies. It's written by my friend Jonathan Losos. Well, it's not too late. You can still read it. And when you do, you'll be amazed at how quickly vertebrate populations can shift their anatomy through evolutionary processes. Well, believe it or not, the fabella is showing up more often in humans. Now you may ask, why? What's it doing for us, and why do we need it now? Need it now. Get it? Need. It's in our knee. Oh, man. Okay, so I've resorted to puns because I don't have a good answer to that question of why it's happening. We're not sure what it does for us, if it does anything at all. But the team that discovered this increase in the incidence of fabellae speculate that maybe a shift in nutrition over the last 100 years that caused the human skeleton to get larger and heavier could be putting the components of the knee joint under more pressure. And that's what's triggering the formation of a fabella. They also found out that if you have osteoarthritis, you're more likely to have a fabella. But we don't know if there's really any cause and effect relationship with that. We just see it happening. So that may or may not be part of the story. As a matter of fact, there are now just a lot of questions that need to be answered. But interesting and fun questions about human anatomic variation, right? I love science. By the way, before we leave this topic and go on to our next puzzler in the realm of anatomic variation, I want to mention something that this story reminds me of. A very strange phenomenon in the world of human anatomy and physiology teaching. Whenever the idea of keeping up with new and updated science content in A&P teaching and in the resources we use for teaching, such as textbooks, I often hear something to the effect that, yeah, physiology probably changes a lot over the years, but anatomy doesn't change. Well, I hear this from students, but I also hear it from A&P teachers. Now, I don't usually agree with folks when they say this to me, but not being a fan of needless contradiction in conversation, I usually do just soft pedal my response by not actually challenging them. But looky here, we have yet another case of human anatomy changing over time, the evolution of human anatomy. Of course, that's not something we see a lot of, but we sure do see a lot of change in our understanding of human anatomy, don't we? As more specimens are studied, as more and different questions are asked, as old ideas are challenged, as our technology improves and we're better able to observe, measure, and explore the structures of the human body, our understanding of human anatomy changes a lot, in my opinion. I think we're better educators if we don't close the door on updates in human anatomy. I think we're going to miss some very important ideas if we do that. Just a thought. The free distribution of this podcast is sponsored by the Master of Science in Human Anatomy and Physiology Instruction, the HAPI degree. I've been on the faculty of this program at Northeastern College of Health Sciences from the beginning, just over 10 years ago. And I'm still excited about all the evidence-based teaching strategies that our learners apply directly to all the major topics in the typical anatomy and physiology course. Check out this online graduate program at northeastcollege.edu/HAPI. That's HAPI, or click the link in the show notes or episode page. You and I talk about macrophages a lot in A&P, right? When we're teaching A&P. They come up a lot. Macrophages are little scavenger cells. Okay, they're not little. They're big. That's where the name macro comes from, right? On a cellular level, they're pretty big cells. And they do phagocytosis, and that gives us the other part of their name, phage, which ultimately can be translated as eating or ingesting or gobbling up. And that's the technical terminology I like to use. Macrophages are the gobbling up cells, and they gobble up damaged cells and cell parts and other debris and particles that we don't want floating around in our body. And so they have a very valuable and important role in the human body, right? Helping keep things clean and helping protect us from infected cells and other kinds of potentially damaging particles. Now, something about macrophages that I don't really dive into much in my A&P course, but you might, and that is where they come from. Macrophages actually have two distinct sources of development. One cell lineage arises from monocytes. And of course, monocytes are immune cells that are derived from the bone marrow, the myeloid tissue. But there's this other set of macrophages that is developing in a different way, that's independent from monocytes. And these macrophages, they come from cells that have spread out into the tissues during embryonic development. And so those macrophages that end up in different tissues, they're sometimes just called tissue macrophages, or they might be called tissue-resident macrophages, or a lot of times they're just named for whatever tissue they're in or whatever job they're doing in that tissue. And that's an important aspect of this, and that is they have different jobs to do in different tissues. And so they're going to express different genes depending on which subset of macrophages they are, which tissue they're in, and what job they're doing. And so it may be something beyond this scavenging of damaged cells and so on, or it may be a different way of doing it or a specific set of things that they're gobbling up. So that's important to know about macrophages. There are different subsets of them. Now, when we're looking at these tissue macrophages, we see that some of them have a role in producing an inflammatory response. But there are other subsets of macrophages that have an anti-inflammatory role. We can't say that macrophages in general promote inflammation, and we can't say that in general they inhibit inflammation. What we can say is, yeah, they're capable of either job, and it just depends on which macrophage you're talking about. Now, something else I want to mention before I even tell you why I'm talking about macrophages is I want to remind you of the disease rheumatoid arthritis. Like, we need reminding of that. That's something that is throughout our human population. We're reminded of it frequently because a lot of people suffer from rheumatoid arthritis. And we're doing a lot of work to try and relieve that suffering and produce effective treatments and someday maybe even a cure for rheumatoid arthritis. And it's an immune disorder that is associated with inflammation of joints. It involves also, because of that inflammation and various processes going on there, with the destruction of cartilage and the destruction of bone in our joints that are affected by the rheumatoid arthritis. Turns out that macrophages have a key role in initiating rheumatoid arthritis. Now, in some recent research, so there it is, what you've been waiting for, like, why is he talking about this now? We know all that. Well, there's some recent research that was looking at a specific subset of macrophages, specifically those that expressed a gene called CX3CR1. And I'll never remember that. I have that written down on a little piece of paper here. CX3CR1. Yeah, okay. But that's a particular gene, and it's one that they were looking at, and they found that these macrophages that express that gene, they are found in the form of a layer of cells. Now, that's kind of weird, isn't it? You always think of macrophages as being these somewhat independent scavengers. They're like cats that, are roaming around independently of each other and gobbling up mice, right? But these CX3CR1, I hope I don't lose this paper. I hope it doesn't drop on the floor. CX3CR1 expressing macrophages, they are found as a layer of cells. And that layer of cells forms a barrier, sort of like this, very thin protective membrane inside a healthy synovial joint. So where they're found in the synovial membrane is beneath that layer of synovial fluid that is being produced by the synovial membrane. So the first thing you'll hit then is this barrier made up of all these macrophages linking themselves together into sort of a membrane. And then beyond that, you're going to find some connective tissue with fibroblasts in there. And an interesting thing about these CX3CR1 macrophages is, I'm just going to call them what the researchers ended up calling them, and that is barrier macrophages. That's a lot easier than CX3CR1. Okay, now it's easy for me to say that I've said it a bunch of times, but I'm still going to call them barrier macrophages. So the barrier macrophages, it turns out when they looked at them very closely in terms of what genes were they expressing besides the CX3CR1, is that they were expressing genes for proteins that are associated with producing tight junctions in epithelial cells that are connecting together to form a membrane. I mean, we all know about tight junctions, right, and what they do. And they form kind of a seal between the adjacent epithelial cells. So why would macrophages be making the proteins needed for tight junctions? Well, maybe it's so that they can form this layer, this barrier that is sort of on the surface of the synovial membrane, adhering to the connective tissue and facing into the synovial fluid. So, that's kind of weird and interesting. And when they looked at this barrier layer, this layer of barrier macrophages, they found that it was really, the term they used is dynamic. It was not just sitting there. It was reacting to things. So in a set of experiments, they induced arthritis in their experimental animals. And when that happened, when they induced arthritis, that barrier layer started remodeling itself. It loosened the interactions between them and those fibroblasts on the inside so that it kind of started peeling away a little bit. Something that happened there is that they started ingesting, gobbling up, and removing some of the inflammatory immune cells, specifically the neutrophils, that, they come in and they accumulate in an inflamed area and then they die. And that happens in arthritis when there's inflammation. They get in there and these macrophages gobble them up. And then they had some animals, they used mice. Their lab mice, they induced arthritis in a group of these mice. They did that while also messing with that layer of barrier macrophages. So they caused the arthritis and they messed up the barrier. And what happened was that those animals got much more severe arthritis than the ones where they didn't mess with the barrier. So when they didn't mess with the barrier kind of peeled back and started gobbling up the neutrophils. But the ones where they messed with that barrier, those barrier macrophages, then the mice got worse arthritis than otherwise. So whatever was going on there with those barrier macrophages got messed up and the animals suffered worse from the arthritis. What they're thinking now is, what's going on there? I mean, this is a new discovery. We never saw that before. That's really interesting. And maybe here is a pathway to finding some better treatments for rheumatoid arthritis or other inflammatory conditions. Let's find out what exactly is going on there. Let's see what is going on during this dynamic process of reducing the effects of inflammation in arthritis. There's a little bit something different that I never knew about synovial membrane. None of us ever knew it before. And I'm not saying we need to go into that much detail when we're talking about synovial membranes or mentioning arthritis in our course. But it's another one of those things that we can just kind of drop in there and say, this is still an active area of research. We don't know everything there is to know about how these membranes of the body are built and how they function and how they interact with other kinds of tissues that may be discussed, outside of our usual discussion of joints and membranes. Marketing support for this podcast is provided by HAPS, the Human Anatomy and Physiology Society, promoting excellence in the teaching of human anatomy and physiology for over 30 years. Looking for teaching tips and articles outlining research about what works in teaching and learning anatomy and physiology? Well, you need to check out the HAPS Educator, available online at theapprofessor.org/HAPS. That's HAPS. Did you see or hear any of those headlines last week that a new organ was discovered in the human body? It's called the interstitium, and it's made up of little fluid spaces in the connective tissues in the wall of hollow organs like the submucosa, and it's found in the fascia of muscles and in the skin and lots of other places in the body. And I don't know if you're thinking what I'm thinking about these, but don't we already know that there's an interstitium? Don't we already know that interstitial fluid forms little pockets within some of the connective tissues of the submucosa and other areas of the body? And not only that, even if we had not known that before, would discovering them and identify them, would it constitute an organ? Really? Isn't it part of an organ, really a structure within a tissue that makes up an organ? I'm not the only one that has had that reaction. It turns out a lot of scientists are pushing back and saying, wait a minute, this is not new information, at least not in the way that it's being portrayed in the headlines and even by the authors, that this is a new organ discovered in the human body. That's taking it kind of way beyond what they're really seeing here. We've seen those pools of fluid, of interstitial fluid, for a long time. We know that interstitial fluid forms these little pockets. We know that eventually it drains into the lymphatic system, into the lymphatic capillaries, and makes its way back to the bloodstream eventually. What's new here? Well, they did some interesting visualizations. They used a kind of microscopy called endomicroscopy, where they take an endoscope and use that as a microscope, and they can make images down to a depth of about 60 or 70 microns beyond the surface that the endoscope is at. And so they did that and some other interesting imaging techniques and so on, and pointed out that, when we're looking at histology slides in our course, for example, or in a textbook, the collagen bundles look denser than they would in life because these pools drain out and collapse. And of course, we've known that for a long time. But it is, I think, important to go back and really think about all of those little fluid spaces being there and the function that they might have. There is a growing awareness of all the different kinds of mechanical and chemical and even electrical interactions in extracellular matrix and tissues throughout the body. And I think that this article contributes to that overall conversation that we're having and increasing awareness of the dynamic nature of these tissues, which not all that long ago were thought of to be more or less glue or packing material or reinforcement material in the body, and not really an active dynamic area that is involved in so many different functions. It's important for homeostatic balance in our body. So, I do have the link to this article. It's an open article, open access article. I have a link to it in the show notes and on the episode page at theapprofessor.org. I also have a diagram at the episode page. And I also have some other articles, including an article that rebuts, the premise that this is a new discovery and that it's even an organ. Don't you just love those moments in our career teaching A&P where we run across some information that's been out there for a little while that seems pretty basic and pretty important to our understanding of human structure and function, and we somehow missed it? I don't know about you, but I'm thinking, oh my gosh, everybody else knows this, but it just went right past me and I didn't even see it. And now I'm seeing it. And how could I not have known that? And I wish I had known this a long time ago. And I just ran across some information like that. But, we all have those kinds of moments, right? Because how can anyone possibly keep up with this huge explosion of knowledge about human biology? There's just too much out there to keep up with. So we do the best we can. And so I don't feel bad about this. I feel kind of excited about the fact that I made this discovery. And maybe you already know this, maybe you don't, but I'm going to share it with you because I think it's kind of fun. And that is the idea that there are junctions in between the endothelial cells that make up the wall of the lymphatic capillaries that are a little bit different than I ever imagined them. And this was first proposed and demonstrated back in 2007. So it's been out there for a while. And they're still working on, trying to iron out all the details of this. But we already know that the endothelial cells of the lymphatic capillaries are a little loosey-goosey, right? They actually overlap one another a little bit rather than being attached necessarily tightly end to end. And when they overlap a little bit, they form like little microscopic valves that allows fluid from the tissues to push into the lumen of the lymphatic capillary. And then it becomes lymph, and it travels the rest of the way through the lymphatic system and eventually is returned to the blood supply. That part we already knew. What I didn't know is that in 2007, we outlined the idea that there are certain types of junctions that allow that to occur. And they're called button junctions and zipper junctions. And they're actually called that. I mean, can you believe that that somebody didn't get out their Latin and Greek dictionaries and try to put together a multi-syllable word for these junctions? But they didn't, because the button junctions, they're like buttons. And these are the kind of junctions we would typically see in the adult lymphatic capillaries. The way they work is just like the button on a shirt. So imagine, or maybe you don't have to imagine, that you're wearing a shirt right now that is buttoned up the front. And imagine that the left side of your shirt and the right side of your shirt are adjacent endothelial cells that are forming the wall of the lymphatic capillary. And they overlap a little bit, right? Just like they would in a lymphatic capillary. But they can't be totally loose, right? Because it would just fall apart. They have to be held together at some point, and they are. They're held together by buttons. But think about the gaps between the buttons. Right now, try to slide a finger or two, or maybe part of your hand, in between two of the buttons on your shirt. And you can do that very easily, right? So imagine that that is fluid that is traveling from the interstitial space of the tissue, that's tissue fluid, that is traveling into the lumen of the lymphatic capillary, and now it's becoming lymphatic fluid. And that's how it works. So that little flap there that is formed between the buttons, that's your lymphatic capillary valve that is allowing the fluid to come into the lumen of the lymphatic capillary in this case. And there are junctions just like those buttons made up of various proteins, and they're still kind of working out how they're built and what they're made of and so on. But they're the same kinds of materials that you would find with other cellular junctions, but this is a unique kind of cellular junction. Now, what about zipper junctions? Those are junctions that the evidence so far indicates occur only during embryonic development. It seems that our lymphatic capillaries are mostly zipped up during embryonic development, at least certain stages of embryonic development, and then they eventually unzip, or the zippers sort of dissolve and they're sort of reconstituted. We would call this a case of plasticity, where the cell is changing things around and changing the connections from buttons to a zipper. Now think about how a zipper is built. Those connections between the two sides of the zipper, they're made up of all kinds of little tiny elements that hook together. So therefore, it is a much more continuous junction compared to when we have buttons. So imagine you have a shirt or a jacket or something that is zippered up the front instead of having buttons. Now you can't easily slip your finger or much of anything in between those two sides of your garment, like you could with the buttoned up garment. And so that's preventing things from getting through. So it's important that embryologically we go through that step of changing from the zippered form to the button form. And the reason I ran across it recently was because there was a new study from some researchers at Yale that sort of accidentally, they were trying to produce some laboratory mice that were obese so they could just do some studies on it. And what they did was they accidentally produced some mice whose lacteals, the lymphatic capillaries in the intestinal villi, were zipped up instead of having buttons. So that meant that the chylomicrons that should have been absorbed from the gut and into the lymphatic pathway weren't being absorbed. And so instead of ending up with obese mice, they ended up with mice that could not become obese because they weren't absorbing fats that were in their diet. So they were giving them more and more fat in their diet, and they just weren't getting fat. And then they figured out what was going on. So they're thinking knowing about this might help control the condition in people who are already obese, and maybe we can sort of shut down their absorption of fats for at least a little while until they're in a healthier range. I think I'm going to do this as a demonstration in my class from now on. I'm going to demonstrate using a shirt. Maybe I can take a shirt and a zippered garment and put them on the torso model from the lab and demonstrate that. Actually, the students themselves can do that if they have a buttoned or zippered garment on in class, and they can do that themselves and understand how the lymphatic capillaries work, including the lacteals. And you might want to bring up this new information if you want. That might be a good opportunity to stimulate some critical thinking. Like, okay, let's say we come up with a drug that can convert, or maybe some kind of gene therapy or something that can convert the button form to the zipper form of connection in the lacteals. Would that really reduce a person's weight? Well, let's say it does, but what other consequences might that have? Could there be some unwanted side effects in that case? I think there probably could. That's not a done deal yet. And so that kind of gets them thinking, not only thinking critically, but thinking scientifically, which is a form of critical thinking, right? There you go. Something new I learned. Maybe it's new for you too. A couple of weeks ago, I ran across a headline that stated, "Researchers discover new microorgan in human immune system." And you know me, I'm always attracted to headlines that claim that there's a new organ in the human body because, well, sometimes there is. And what really intrigued me about this one is they didn't really call it an organ. They called it a microorgan. And I don't even know what that is. So I had to read the article. And I'm glad I did because it was referring to a piece that came out on August 22nd in Nature Communications. And it's entitled, "Memory B cells are reactivated in subcapsular proliferative foci of lymph nodes." And basically what it described was some experiments in which they demonstrated that memory B cells that are produced during and after an infection in an area get trapped in the nearby lymph nodes, which is not surprising, and they hang out there, which is also not surprising, but they form a focus, a location, that is distinct from the germinal center of the lymph node. It's distinct both functionally and structurally from the germinal center. And these folks decided to call it a subcapsular proliferative focus, or plural would be foci or foci. It's subcapsular because it's just inside the capsule, just deep to the capsule, which forms the wall of the lymph node. And it's proliferative because it's made up of memory B cells, which we know, when triggered, will proliferate and form groups of more memory B cells and some plasma cells, which are going to be actively producing antibodies against the antigen that triggered the formation of those memory B cells to begin with. It's called a focus because it's a location, right? These SPFs, which is a lot easier to say than subcapsular proliferative foci or foci. So the SPFs, that's what's new. And the authors, I couldn't find anywhere in there where they called it a microorgan. Maybe I just missed it. But the point is that we'd never really seen it before, never really noticed it before. And it's really interesting that when these memory B cells, they hang out there, which makes sense they'd hang out in the lymph node, but at a particular location just inside the capsule where they are most likely to encounter the antigen that they're programmed for, it would be right there in the outer area, not deep in the germinal center. So it makes sense that it would be there, right? And it also makes sure that it's a good place for the macrophages, which are acting as antigen-presenting cells, that they would be, able to get to those memory B cells very easily. And not only are the memory B cells hanging out there, but there are a bunch of helper T cells and presumably other cells that are going to be involved in this whole process of getting those memory B cells activated and proliferating and producing the plasma cells, which are going to be producing the antibodies. And all of this together means that process of this secondary response to the antigen, the original antigen, is going to be that much more efficient. And now we're seeing how it gets to be so efficient. And the authors of this communication propose that knowing about this may help us do vaccines better. Because isn't that what vaccines are doing is giving us that original antigen exposure and getting those memory B cells in place. Now we know where their place is. At least one of the places is that they're going to be ready at a moment's notice to mount that very strong and very rapid secondary response, which is the whole point of doing a vaccine, right? Some interesting things in the immune system these days. I have more discoveries in human anatomy to discuss, but before I do, I want to mention something called the A&P Professor Science and Education Updates newsletter. It's a free digital subscription that you can get by going to the approfessor.org/updates. And it includes, well, as the name implies, headlines from all over the world having to do with human science of interest to faculty who teach anatomy and physiology. And there are also education updates with teaching and learning ideas and concepts and news from the world of education. And there are a few updates from the world of the A&P Professor. So if you want to subscribe to that, or even just check out what some of the past issues look like, go to the approfessor.org/updates. I was reading through some articles from New Scientist, and I got to tell you, when I see an article that begins with the statement, "It's time to rewrite the anatomy books," I'm going to read that article. And I'm glad I read this one because there's a very interesting new discovery about a potential new kind of blood vessel that's been discovered in bones. Whether it's really going to require a rewrite of anatomy books is yet to be seen, but this is very interesting early information that you probably ought to take a look at because I think there is something to it. You probably already know that the blood supply of bones usually comes into the bone by way of some arteries at either end of a long bone or maybe near the middle of a long bone, and it penetrates through the cortex or the cortical layers of bone in all the way down to the medullary cavity. And then from there, it radiates back to the outer areas of the bone. And what these researchers that wrote a recent article in Nature Metabolism did was they were looking in mice, and they saw that by using some chemicals on some long bones of a mouse, they were able to make the bone transparent, and they could see the tiny little capillaries that were present in the bone. And they found a bunch that they did not expect that were crossing the bone shaft perpendicularly. In other words, they were going right across those cortical layers and right down toward the medullary cavity. So they weren't coming in a large artery and then going through this whole network of capillaries and so on within the bone and then draining back out through a large vein or maybe 2 or 3 or whatever large veins out of that long bone. They were actually coming in, sort of like seeping in through the wall of the bone. But of course, they're going through these little capillaries. And the same thing for blood escaping back from inside the bone and getting back into the venous circulation back toward the heart. And so they named these newly discovered very tiny vessels transcortical vessels, or TCVs. And then, of course, one of the first things they did, which I would do, is look in human bone and see, and they did find some in human bones. But there's a lot more yet to be done with not only the mouse model, but also in doing human investigation to see whether this is just a fluke or it's a mistake in how they were applying the technology or a mistake in how they recorded their observations or what. But it's looking pretty good so far that most of the blood, and by the way, in the journal article, they propose that over 80% of the arterial blood flow coming into the bone and about 59% of the venous blood flowing out of the bone goes through these TCVs and not through those large arteries and veins that we thought were the only way in and out of a bone. This could really be a boon for medical illustrators who are going to have to draw new pictures of the blood supply of bones. And I think it's just another one of those interesting stories on our journey to understanding the anatomy of the human body. Can you believe it? Yet another previously undiscovered new organ has been discovered in the human body. This time, it's the tubarial salivary gland, which its discoverers would like to see considered to be one of the major salivary organs, along with the parotid, submaxillary, and submandibular pairs, rather than as simply another set of the huge number of minor salivary glands that are scattered all around the mouth and throat regions. The name tubarial comes from its location, or I mean their location, because it is a paired set. So the location is along the torus tubarius. And yeah, I wasn't sure what that is either when I first heard it. A torus is basically a classic donut shape, and that's kind of the shape formed by the cartilage supporting the opening of the auditory tube into the nasopharynx. The pharyngeal mucosa then has this rounded bump surrounding the tube's opening, looking kind of like there's a little donut under it. That is what the torus tubarius is. So, yeah, okay, we're miniaturized and in the nasopharynx, looking directly at the opening of the auditory tube, and embedded in that tubarial mucosa and extending inferiorly from there are a whole bunch of exocrine glands producing a mucus secretion that's coming out of a whole bunch of little ducts. The image in the show notes in your podcast app or at the episode page shows this structure clearly if you can't imagine it. That image looks almost as if somebody had been miniaturized and took a snapshot with their mobile phone. And mark my words, someday that's going to be a thing. Now, I'm always skeptical of discoveries of new major organs, so I'm still kind of taking a wait and see attitude, at least a little bit. Yes, the discoverers did look at many, many individuals and made all kinds of other observations that back up their claim. To a point, but I'm thinking maybe we need a little bit more. I'm not sure. And of course, we need time for science to do its job and let folks replicate and debate and mull over what's going on. A question I have is that there are multiple ducts observed in the tubarial salivary gland, and I'm not sure, therefore, how it can be distinguished from groupings of the many minor salivary glands we've already established in anatomy. On the other hand, the sublingual salivary glands have multiple ducts, so I guess the fact that there's a bunch of ducts doesn't exactly push the tubarial salivary glands off the list. Any list of what's an organ and what's not an organ, or what's a major organ and what's not a major organ and it's a minor organ, is only partly a matter of science theory. It's also partly a matter of our need to figure out what label we want to put on structures. Those labels are there to help us understand the body better and also to communicate more accurately with each other. So what the community of scientists needs to do now is to figure out whether this newly proposed name and newly proposed designation as a major salivary gland rather than a minor salivary gland is useful or not. Now, I don't have a real strong opinion one way or the other about this, just questions. A big question is why insist on calling it an organ? In reading the paper, you'll see that the author's primary interest in doing so has to do with the clinical implications. If it's one of the major salivary glands, then it's worth putting on the list of structures that we want to take steps to spare or protect during radiation therapy, And that's where these folks are coming from the radiation therapy angle. If the tubarial salivary gland is just a random set of tiny glands we imagine to be scattered all over the place, well, then maybe sparing them is impractical. But I think the authors are saying, hey, look at these images. These glands are concentrated in a rather defined area, and there are a lot of them. And we can take steps to avoid damaging them now that we know where they are and that they're even there. And patients will be better off for doing that. So here they are, and here's what we're calling them because they need a name to put on our list of things to consider when planning radiotherapy for a patient. So, when you look at it from that angle, maybe calling the structure an organ is not such a bad idea. I don't know. As usual, some folks are calling for a rewriting of textbooks. Well, I always want to hold on a minute when I hear that, because I hear that a lot. Maybe my ears are sensitized to that. What? Something needs to go into a textbook? And a lot of times it's something that's just way too early to go into a textbook. Something that's going to be soon disproven or at least modified in some way. And the other thing is that, especially for undergraduate A&P textbooks, we have to ask the question, is this really a detail that our students really need to know? Even at an advanced level, like in a medical anatomy course, there are some things that maybe are just too much and maybe don't have a clinical or practical application. I think this one does, but I'm just saying in general, those are some questions that have to be asked before it actually gets into textbooks. Things don't just automatically flow from journals into a textbook. There's all kinds of editorial decisions to be made, and scientific questions and decisions and observations need to be made. So are we really ready for that at this moment? I don't know. Maybe science has to do its work a little bit more so that we have greater confidence in this idea. Or maybe it is time now. But no matter what, I'm also thinking it's a great time to mention it to our students. Even if it's outside the official content of our course, that is, maybe even if we're not going to put it on the test, make it into a test item, maybe we want to discuss how science works, and this is a good opportunity to do that. And it's also a good opportunity to discuss that we don't already know everything. And it's a good opportunity to discuss what makes an organ, which is a lot fuzzier than a lot of students imagine. And well, there's all kinds of interesting things that can come out of such a discussion, right? Hey, why not share this episode with a colleague and get their thoughts on it? It's easy to do. Just go to the approfessor.org/refer to get a personalized share link that will get your friend all set up. I always provide links if you want to know more about the topics of this episode. If you don't see links in your podcast player, go to the show notes at the episode page at the approfessor.org/podcast. And while you're there, you can claim your digital credential for listening to this episode. And you're always encouraged to call in with your questions, your comments, and ideas at the podcast hotline. That's 1-833-LION-DEN or 1-833-546-6336. Or send a recording or written message to podcast@theapprofessor.org. You're invited to join my private A&P teaching community at the approfessor.org/community. I'll see you down the road.
The A&P Professor is hosted by Dr. Kevin Patton, an award-winning professor and textbook author in human anatomy and physiology.
Each episode of this podcast is no deposit, no return.